This is the second recording of two videos explaining resting potential and action potential. In the previous one we concentrated on the resting potential, and now we are looking at the action potential, how this wave of depolarization moves down an axon. There will be a third video in this series which will look at synapses and how a nervous impulse might be passed from one neuron to another. This is our summary sheet of the resting potential and where we ended last time's video, the last video. Now, moving on from there. That is how the pump and the leaks work, our Na plus K plus pump and our K plus leak channels. So how does that translate into an action potential? Well, we get to, so I'll get my cursor on the screen, our resting potential along here. Now, this graph, it should be noted, shows you the voltage across a particular piece of membrane. This is not the charge moving along the membrane. This is a shot of one piece of membrane over a period of time. We've got time on the x-axis, and at the start of this time period, that is at minus 70 millivolts. It is at resting potential. Now, important and exciting things start to happen when something happens to raise our voltage to the threshold value. That is about minus 55, 57 millivolts. And that might be the arrival of an action potential from somewhere else. It might be something causing ions to flow one way or another. But one way or another, we need to reach that threshold value for anything to happen to change us from being just a resting potential. Oh, there's our fancy animation. Now this comes to gates and how these can affect that membrane potential. There are a number of gates. These refer to protein channels for facilitated diffusion. We can have mechanically gated gates. This would be, for example, in Pacinian corpuscles. Pacinian corpuscles are sensors which detect pressure changes. As the Pacinian corpuscle will be pressed down, it opens up these mechanically gated channels, and that allows Na plus ions to flood in. We've got ligand-gated channels here. On this example here, we've got a ligand, i.e. something that binds, coming on and binding to this gate, and then opening up this channel. In this case, maybe for Na plus ions. This might be a neurotransmitter released from a presynaptic membrane, coming along and binding onto this channel here, this ion channel here, opening it up, allowing ions to flow through. This would be doing the same thing, but with an intracellular ligand, something from on the cytosolic side, coming along and binding and opening it up from the inside. I guess an opposite version of this being when ATP, a rise of ATP in beta cells, closes K plus leak channels. But then we get onto our voltage-gated channels, and these are the ones we're going to focus on in this video when we're thinking about action potentials. Now, when you are at resting potential, i.e. with a strongly negative charge on the inside of the membrane and a strongly positive charge on the outside, that configures this protein ion channel to be closed. It closes this gate. So how's this going to happen? You will have various R groups on our protein controlling its tertiary structure. Those R groups will, some of them have different charges, and they will be at different charges according to the voltage they're at. If you change the voltage, that changes the charge on certain key R groups, and that leads to the gate opening and the channel becoming open. Here we've changed the charge from being very negative to slightly negative, and that opens up this gate. It should be said that all of these are for facilitated diffusion. And a little bit of revision on facilitated diffusion. It is protein-mediated transport. It is substrate-specific. It will be based on size and charge and shape. and movement by facilitated diffusion will be governed by Fick's law, or if you prefer, Fick's law describes how quickly facilitated diffusion occurs. So our limiting factors will be, or rather our factors affecting it will be surface area. Larger surface area makes it happen faster. Larger concentration gradient will make it happen faster, and a larger diffusion distance will make it happen more slowly. As temperature rises, you would expect the rate of diffusion by facilitated diffusion to rise, but in facilitated diffusion that is only up to a certain point, because at a very high temperature the proteins concerned will denature, and that will stop facilitated diffusion dead in its tracks. Well, let's look at how look at that rotation. Let's look at how all this is relevant to us and our action potential. Now we're at our resting potential, and then something happens to raise our membrane potential to above the threshold value. What that does is it opens up voltage-gated sodium ion channels, and get that phrase into your mind. That's a great phrase to use. Voltage-gated sodium ion channels. Each one of those words is important. They're not just sodium channels. We don't have sodium metal going around in our cells, in our extracellular fluid. That would be dangerous. And they are voltage-gated. So the membrane potential rises, we get over threshold value, and this is what happens. We've got sodium ions at a low concentration inside the cell, and so we open up our voltage-gated channel, and what's going to happen? Na plus floods in by facilitated diffusion down that concentration gradient, that electrochemical gradient. Because these sodium ions carry a positive charge, this flips the voltage over. Whereas previously we were negative on the inside and positive on the outside, this influx, this rapid influx of Na+, these positive ions, reverses that polarity, making it positive on the inside and negative on the outside. The word we use to describe this is depolarizes. The membrane becomes depolarized. This process is called depolarization. Another key phrase to use is the phrase all or nothing response. If your sodium ion channels open, they open, and that will rapidly depolarize the membrane. You can't have a half depolarized membrane. Either the channels open or they don't. Well, let's see how that plays out on our graph. And you'll see we've got an illustration here of three forms of this Na plus voltage-gated ion channel. We start here with this one. We're at resting potential, highly negative on the inside, highly positive on the outside, and it is closed. There we go, at resting potential. Now something happens to change that a little bit, to get us over that threshold value, and that causes the Na plus the voltage-gated Na plus channels to open up, meaning that Na plus floods in. Now that isn't going to last forever, and after a certain period of time, and particularly as it becomes more and more charged, these channels here go from a state of being open to being closed and inactivated. So we have three stages: closed but openable, closed but, if you like, active, going to open, and then these open ones going to inactivated here. At this stage we get to the point whereby our voltage-gated sodium ion channels are closed, but not only are they closed, they are inactivated. That means they cannot open again for the time being. They are closed and unopenable. That is in contrast to these ones here, which are closed and openable. And it just takes a very short amount of time, but that time is going to be significant, and we'll find out about that when we think about the refractory period later. It takes an amount of time for this protein here to be reconfigured from this inactivated and closed state to the active and closed state at this point. Let's look at this as an animation. This is, we'll go this one. Here are our gates here. We've got the Na plus ion channel gate there and the potassium channel there. We'll have a look at that in just a moment as well. So we'll begin depolarization. We're along our resting state. We get to threshold potential, the channel opens, and then the gate closes at the peak. It's now closed, it won't open up again. We're back to our resting potential now, and here we go, it's ready to fire again. Well, that's great. Now we're going to start looking over here at this potassium ion channel there. Now this would normally be closed. This is different from the leak potassium ion leak channel. This is a voltage gated potassium ion channel, and it will open when voltage becomes very positive on the inside of the membrane. As it opens up, so we'll let the sodium ion channel do its stuff, and then as this opens up, K plus exits, is allowed to exit by facilitated diffusion, and that will bring our voltage on the cytosolic side of the membrane back down again as positive ions leave the cell. Do have a look at that in your own time. You can obviously see the website reference there. So back onto this. Now we're going to look at it at various stages along the axon, and we're going to think about how the action potential is propagated down the axon. We have, if my cursor come up, there we go. We have our resting axon here, and this is at resting potential, negative on the inside, positive on the outside. Well, let's have a look. We have then a region, let's not explain why we have this region of depolarization here, but let's say we have it. This could be an arrival of an action potential from somewhere up here. We now have a negative charge next to a positive charge, next to a negative charge, next to a positive charge. And so we have the opportunity for a small local current. Now this small local current will take this negative charge here and make it slightly less negative. We have charge moving from this positive to this negative here, or more accurately from this positive to this negative here, as positive ions flow this way or electrons flow that way, with positive ions flowing this way, and that will make that less negative. Now think back to what we've learned about our voltage gated sodium ion channels. Once you get to a threshold voltage, so about minus 55 millivolts or above, those voltage gated sodium ion channels are going to open. Now as they open up, they are going to totally depolarize that membrane, and then where's my cursor gone? There it is. Then it will all become depolarized, and we are pushing our action potential down the neuron. And see, we've pushed it. We've started off here, it's got to here, and now it's going to there at the bottom. Well, what happens behind the action potential? That's the next important thing. Well, behind it is repolarization. During repolarization, two things are occurring. Number one, our sodium ion channels become inactive. That's as we discussed before, looking at the shapes of the gates. And also because our voltage-gated potassium ion channels open. I seem to have left out the word ion in these. You'll have to forgive me. As those potassium ion channels open, the potassium ions flood out of the cell, and that repolarizes it. That brings us back to resting potential. In fact, it pushes us past resting potential. There is overshoot, and that is called, you may want to write this word down, that is called hyperpolarization. And we'll look at that in a couple of slides' time. The sum effect is to repolarize the membrane. with a negative charge on the inside. It's also to give the membrane time to repolarize while our voltage-gated sodium ion channels reconfigure from the inactive to the active state. Let's have a look at this again. I'll go to this one this time. So we're in an unmyelinated axon. We'll think about the myelinated one in due course. Again, do look at this animation in your own time. Let's have a look. And as we click on this, look at the local currents it generates. So we have threshold value achieved, Na plus ion channels open. We have repolarization behind it, and that little arrow pointing to the refractory period is showing you that the time before which you cannot have another action potential. And we'll have a look at that just presently. So back onto here. Now we're thinking about the propagation down the axon, but I also just wanted to, on this slide, give you a little bit of history of this. The initial experiments were done on the giant axon of a squid. Well, why? Were they particularly available? Were there lots of squid around at the time? Well, there are still lots of squid around now. There always have been a lot of squid around, but I digress. The giant axon of a squid is huge. It's the axon which goes from its tip down towards its base, and it controls the siphon that a squid has, which allows it to squeeze water out very quickly to give it jet propulsion to escape from a predator, maybe a sperm whale or something scary like that, which has come along to gobble it up. And therefore it has to be a very fast response, and therefore it has to be huge. And it is huge, and we'll explain why exactly in terms of speed later on in a few slides' time. But these experiments were done by Alan Hodgkin and Andrew Huxley, and they're all very clever, and we applaud them for it. On to the refractory period. So we're now thinking about this phase of the action potential between the peak of the peak voltage and the trough of voltage. What is the significance of this time here? The time taken for it to get back to resting potential. During this time, what is happening? Well, the membrane is repolarizing. That is due to the potassium ion channels opening and the sodium ion channels closing. And more to the point, the sodium ion channels closing and still remaining inactive. What is the purpose of this refractory period? It makes sure the action potential can't go in both directions down a neuron. Now, if a neuron, if you artificially stimulate a neuron in the middle of it, it will happily conduct an action potential in both directions. It's quite capable of doing that. And so you don't want that to happen within your nervous system. If it does, then your nervous system just becomes a great big mass of noise. And so this is one of the two things which stops action potentials going in both directions. The other being the synapse, and we'll look at that in the third video. And here are some text explaining, putting into words really, just what we've been talking about. And so you'll have this text on the PowerPoint handout that you will have printed out. Great. So key point, I guess the final bullet point here, the time, i.e. the refractory period, that time is long enough to stop the impulse passing in both directions, as by the time the ion channels, that's the sodium ion channels, are ready to open again, i.e. they've come out of their inactive state and gone into their active but closed state, well, by then the membrane is back at that resting potential level. The speed of action potentials. Well, there are a number of things that affect this. Number one, temperature. Remember, these action potentials ultimately are generated by facilitated diffusion, by the diffusion of Na plus ions or by the facilitated diffusion of K plus ions in order to depolarize and repolarize the membrane. And because it's using it facilitated diffusion. At higher temperatures, particles have more kinetic energy, and therefore these ions will diffuse more quickly through those channels at a higher temperature. But naturally, if the temperature gets too high, your protein channels are going to become denatured, and therefore conduction will stop entirely. Next factor will be the diameter of them. Well, if you have a wider diameter, just as you would do for a wire, resistance is lowered. And because resistance is lowered, you will get, in a wider direction, less resistance to small local currents. And therefore, these, number one, these local currents can travel a little bit further. And also, they are, well, they're not faster, but they are, yes, they're likely to set off your action potential just slightly further away from where it is. And that speeds everything up. Wider diameter, less resistance to the small local currents. And finally, myelination. You will remember from GCSE the myelin sheath, or the fatty sheath which surrounds the axon, and we've described that in the past as insulating the axon. Well, that remains true. That doesn't change at A2. They insulate the axon, and we'll look in detail as to how they do this in a process called saltatory conduction. The long and short of it is that myelinated neurons are much faster, or rather they conduct action potentials much more quickly than non-myelinated ones. And if we just flick back a couple of slides to I've gone too far. There we go. To this, talking about the giant axon of the squid. Squid have these giant axons because they are invertebrates. And only vertebrates have myelinated axons. And as such, therefore, this squid needs a very large axon to compensate for its lack of myelin, to make sure this is quick. And therefore, experimentally, and this is the key for it all, experimentally, it's much easier to get your electrode into the giant axon of a squid. To get this inside a large axon is much easier than getting inside a small axon. Now, these days, you can get them into very tiny axons. So if you're doing developmental work on Drosophila melanogaster, that is a little fruit fly, which is very often used in genetics. It's a model organism in genetics. You will need to get that electrode into a very tiny axon, get it inside what is a very small cell. Indeed, that axon may be the whole fly may be smaller at some stage of its larval development, at the kind of stages you'd be doing this in if you're studying its development, as the whole axon of the giant axon of the squid, the fly may be smaller than that axon. But modern techniques are better. At this time, this is the first type of experiment done on it. It's done on the giant axon of the squid because they're big. Right, I have to flick back through. Here we go. Forgive me. Saltatory conduction. Let's start off by looking at how this myelin is formed. It's formed by a Schwann cell. And a Schwann cell is a cell with a nucleus. And what it does is it wraps itself around the axon. We're looking at it as a cross-section here. And as it wraps round, you can see the axon, the arrows are slightly wrong, that should go to there and then on to there. You can see that it's building up here layers of membrane wrapping around this axon. And as these layers of membrane build up, so you get this sort of effect. Hopefully you can see on here, this is the axon, and here, I don't know if you can make out on this picture, you can here. Do you see here? These are little bits of membrane, and here as well. And this black mass here is just membrane, layers and layers of membrane sandwiched on top of each other. There's the nucleus of the Schwann cell there, and this is myelin. And myelin is essentially lots of layers of cell membrane, so it's lots of phospholipids and a fair bit of protein as well, because that is the constituent. Those are the constituents of cell membranes, and altogether we call that the myelin sheath. Another perspective on it. here's our axon here, and this is the Schwann cell having wrapped itself around, giving us these layers of myelin, forming this myelin sheath. Now, the whole axon isn't insulated in this way. It has nodes of Ranvier. Now, you can decide how to pronounce this word here. You might call it Ranvier, you might call it Ranvier. I like to say it a little bit like a motorcycle revving its engine. Ranvier, something along those lines. You take your choice on that one. And the distance between these nodes of Ranvier is 1 millimeter. Now, that is a significant distance, and we'll get onto that distance in a little bit. What that allows to happen is saltatory conduction. That means literally jumping conduction. The myelin insulates the axon and prevents ion movement. So here you can't get ion movement at this point. It won't happen. You don't have the ion channels there, and even if you did, they'd be wrapped around with myelin, and it wouldn't be able to. The ions wouldn't be able to move. However, what the current can do, where's my cursor gone? There it is. What the current can do is it can flow the short distance between the nodes of Ranvier, and therefore an action potential here, just have a look at the charges, negative, positive, negative, positive. It can conduct, you can set up a circuit here around, looping around the myelin, from one node of Ranvier to the next node of Ranvier. At some point I'll grow tired of saying Ranvier in that way. And what this will do is it essentially means the action potential leaps from one node of Ranvier to the next. Can't travel backwards, we still have a refractory period in action. See here, this is our repolarization, and so waiting for that to become repolarized. And so it jumps from one to another rather than traveling through the whole of the membrane, and so it becomes much, much faster. In essence, what it's doing is it is making these local currents, the local currents we looked at in a few slides back with normal action potential conduction, it's making those local currents longer. And in making them longer, it's making the conduction itself much, much faster. In fact, because you're setting up a local current here, as the action potential is traveling from this point to this point, it will move at the speed at which a current is set up, and currents are set up at the speed of light. So we have kind of fast conduction and slow conduction. Right, where's my cursor gone? Let's have a look at that in action. Here we go. Begin saltatory conduction. Action potential there. Speed of light. Really quick. Here we go. And speed of light. There we go. Let's do that again. It was so much fun. We've got our action potential there normally. Oh, speed of light between that and that. And here we go. So back onto this. Another significant point of this, because it doesn't have to travel through all of the membrane, fewer ions are allowed to move by facilitated diffusion, generating our action potential. So fewer Na plus will diffuse in, fewer K plus will diffuse out, and therefore you require less ATP in order to redress the balance with your Na plus K plus pump. So less ATP is used, and the thing goes faster. All in all, a bit of a winner for vertebrates. All in all, a full-on winner. We said we'd mention this distance here of 1 millimeter. What's the significance of that distance? The significance is that is the maximum distance whereby this potential difference, allowing for resistance, is large enough for an action potential here to stimulate an action potential over here. If this distance were longer, actually, by the time you take resistance into account, you don't get enough of a potential difference to get this part of the membrane above threshold value, and therefore natural selection has given us the perfect distance, 1 millimeter. Long enough for fast conduction, short enough for conduction to occur at a reliable enough voltage. And so we come to a sample question and answer. If I were you, I would pause the video at this point and look at this question. How is an action potential propagated down an axon? I'm about to reveal the answer, so get ready on that pause button and pause now until you have written down your answer. I'm now going to give the big reveal. Here we go. You may also think of other points to make along with these ones, but I think that will do you well. Thank you.